Hi friends, welcome to MedClarity. So today we are going to see about the histology of the serous salivary gland. So before that, we need to know what is digestive system. So it's the system which is involved in digestion and metabolism. And if you see the digestive system, it is divided into three divisions. The first division is the oral cavity. Second division is the gastrointestinal tract. Third division is associated digestive organs. So in the oral cavity, it starts from the tooth, lip, tongue and salivary gland. So you can remember the mnemonic TLTS. And second division is the gastrointestinal tract. It starts from the esophagus, stomach, small intestine, large intestine, appendix, and finally the anal canal. So the third division of the digestive system is an associative digestive organs, which includes liver, gallbladder, and pancreas. So today we are going to see about the histology of the serous salivary gland. So first of all, what is salivary gland? The gland which secretes the saliva is called as a salivary gland. If you see the salivary gland, it's divided into two types: major salivary gland, minor salivary gland. So the major salivary glands are parotid gland, submandibular gland, sublingual salivary gland. And the minor salivary glands, it is located in the cheeks, lips and tongue. So coming to the major salivary gland, so parotid is the largest serous salivary gland. And location, it is located in front of the tragus, just lateral to the ramus of the mandible. Finally, it drains into Stenson's duct. And coming to the submandibular gland, it is located at the floor of the mouth, and it's the second largest salivary gland. So this finally drains into the Warden's duct. And the third one is the smaller salivary gland, that is in sublingual salivary gland, which is also located at the floor of the mouth. Finally, it drains into the Warden's duct. And coming to the minor salivary gland, the labial glands are present in the lips, and buccal glands are present in the cheeks. Von Ebner glands are present in the tongue. So all these finally drains into the Warden's duct. So next, before knowing about the histology of the serous salivary gland, we have to know about the general architecture of the salivary gland. So in all the salivary glands, mainly three structures will be present: capsule, and then acini. Third one will be the ducts. So in the capsule will be present outside. From the capsule, the septa, it extends into the gland, which divides into lobes and lobules. And then the acini will be three types: serous acini, mucous acini and mixed acini. And the third structure will be the duct. So in the duct, you can see the intralobular or intercalated duct, striated duct, interlobular duct, interlobar duct, main duct, which finally drains into the oral cavity. So now we are going to see about the serous gland. So the example of the serous gland is the parotid gland. So here in the parotid gland, outside if you see, it is covered by an capsule. And from the capsule, the connective tissue septa, it extends into the glands, which divides into lobes and the lobules. So within the connective tissue septa, you can find the arterioles, venules and interlobar duct. So this is the interlobar duct and this will be the venule, and then you can see the arterioles also. Okay. And then apart from that, you can see the serous gland. So if you see the serous gland, it is pyramidal shape and you can see the rounded nuclei at the center and then a smaller lumen. So initially if you see, it will be rounded structure and it will have one pyramidal cells with the small rounded lumen. If you see the secretory granules, it accumulates at the apical parts of the cell. So because of that, it pushes the rounded central nuclei to the basal part. So the nuclei is pushed to the base. So you can see the rounded nuclei at the base. Then you can see the ducts. So first you can see the intralobular duct or intercalated duct. So this intralobular duct, it merges to form the striated duct. Okay. So here you can see the striated duct, and then striated duct, it merges to form the interlobular duct, and then the interlobular duct finally it forms the interlobar duct, which finally forms the main duct. Then main duct drains into the oral cavity. So within the lobules, you can see a intralobular duct and then striated duct. Apart from that, some of the adipose cells. So coming to the duct, initially the intralobular duct or the intercalated duct will be lined by an cuboidal epithelium. Then when it merges to the striated duct, it is lined by a columnar epithelium, and then finally when it lines the interlobular duct, so this changes gradually from simple columnar epithelium to the stratified columnar or the stratified squamous or stratified cuboidal epithelium. And finally the interlobular duct, it gets converted into one interlobar duct. An interlobar duct finally drains into the main duct. So this interlobar duct, it gets converted into one pseudostratified columnar epithelium. Then finally it drains into the oral cavity. So this much about the histology of the serous gland. So one important function in the serous gland is that surrounding the serous cells, you can find a myoepithelial cells. So these myoepithelial cells mainly it helps in the contraction. So these myoepithelial cells it also contains the pyramidal cells with the basal nuclei. So next we will be seeing about the functions of the serous salivary gland. So the main functions of the serous salivary gland, usually it secretes about 1 liter of saliva in 24 hours. And next, when nerve is stimulated, this myoepithelial cells it contracts. So when myoepithelial cell which is surrounding the serous gland is contracting, it expels the secretions from the serous gland into the intralobulated duct or the intercalated duct. So from the intralobular duct, the secretions it goes to the striated duct. Then from the striated duct it goes to the interlobular duct. From the interlobular duct it goes to the interlobar duct. Then from the interlobar duct it goes to the main Stenson's duct where it drains into the oral cavity. And then coming to the function of the serous gland, if you see the serous gland, it contains the abundant salivary amylase enzyme. So when carbohydrate food enters into the mouth, this amylase is produced by the salivary gland, mainly the serous salivary gland. It initiates the breakdown of carbohydrate food into starch and smaller carbohydrates. And apart from that, the serous cells also secretes bacterial enzyme which is called as lysozyme. So this lysozyme secretion it hydrolyzes or eradicates the cell wall of bacteria and inhibits the microbial growth in the oral cavity. Then coming to the function of the connective tissue. So if you see within the connective tissue of the salivary gland, you can see the plasma cells. So this salivary gland connective tissue, it produces the salivary antibodies which is called as an immunoglobulin A. And apart from that, the serous cells it secretes a protein component, and this protein component it binds and transports into the connective tissue. Then it transports into the saliva. So now the immunoglobulin A antibody which combines with an antigen and it helps in prevention of proliferation of the oral bacteria. And finally coming to the ducts. So you can see here the serous acini. As I have told you already, the serous glands, by the contraction of the myoepithelial cell, it squeezes the secretion into the intercalated or the intralobular duct. Then the secretions goes to the striated duct, and then apart from that, from the striated duct it goes to the intralobular duct. Okay, so here if you see the striated ducts, it mainly acts as an ion transport. So this striated duct reabsorbs the sodium and chloride ions from saliva and releases the potassium and the bicarbonate ions into salivary secretion. So because of that, it results in the hypotonic saliva. And if you see in the striated duct, there will be many infoldings. So these infoldings, it produces the electrolytes and fluids and it produces an energy. So next, coming to the applied histology. So coming to the applied histology, first you are going to see about the tumors of the salivary glands. So tumors of the salivary gland, it is divided into two. The first one is a pleomorphic adenoma. So here in pleomorphic adenoma, there will be a mixed benign appearance of cells. Like epithelial cells, it presents in mucoid matrix, myxoid and chondroid tissues. And second one is a mucoepidermoid carcinoma. So it's a malignant tumor and it's common. So it will have four type of unmixed cells like mucin producing, squamous, intermediate and the clear cells. And next, if you see some of the infections like mumps, so it's a viral infection. And then if there is any radiation given to the glands or when we take any antihistamine, so because of that it can lead to side effects and there is a decreased production of the saliva. So this can cause a dry mouth. So this dry mouth otherwise called as xerostomia. And next is the viral infections like rabies infection. And then apart from that, when there is nausea, the autonomic activity will be stimulated and there will be increased production of the saliva. So this increased production of the saliva, we call it as an sialorrhea. So that much about the histology of the serous salivary gland, functional correlations and applied aspects. Thank you very much.